Today I shall talk to you of a marvelous vaccine in which I was involved in helping our Singapore government in 1983, and also the International Agency for Research in Cancer of the World Health Organization. This is the Hepatitis B virus, one of the most infectious killer diseases ever known to man. In short form, it's called HBV. It was never discovered, even though it was killing many millions of people from the time of Hippocrates in BC 500, or about the same time as Confucius, to today. In 1972, my good friend and colleague, Professor Baruch Blumberg, from the Fox Chase Cancer Institute in Philadelphia, a clinician and a scientist, patiently and together with his colleagues, discovered the virus or the infectious agent that was the killer. It was many years later that working on new reagents that it could detect the hepatitis B virus. In short, the reagent was used to detect HBsAg. Using this reagent, countries from all over the world found that it was very contagious. It was spread through saliva, through blood transfusions, because there were no adequate facilities to screen for the virus at that time. And more than 380 million people were infected globally. Researchers throughout the world, working with new reagents, discovered that this virus was not only present in people with chronic liver disease, acute jaundice, and many who died from it with acute infections, but it was also present in the chronic disease and in liver cancer. Liver cancer then was the top killer and the number one cancer in many countries in the Asia-Pacific region from 1975 to 1985, when large-scale surveys started to be done. It was not till early or late 70s that a primitive or prototype hepatitis B vaccine was made from the blood of people who were carriers of hepatitis B. In very early studies, which was done in New York by the Merck Shop and Dome under its director Maurice Hillman, and by Palmer Beasley in Taiwan, they found that the vaccine was effective in protecting babies born to infectious mothers. However, there was a lot of controversy. Should such a vaccine be used at all? Because it was made from the protein product of the virus, which is known to cause the cancer. It was not until molecular techniques arrived, when DNA analysis of liver cancer tissues and cirrhotic liver tissues showed it was the DNA of the virus that was the cause of the liver disease and the liver cancer, and not the HBsAg particle. It was a very tedious and very difficult technique to make a very safe vaccine for the many millions of doses required for all over the world. In 1983, the Singapore government gave to us, our research group, which is a combined team from the National University of Singapore and the Ministry of Health, scientists and clinicians, to develop a vaccine for human use. Assembling a team of PhD doctors, biochemists, pathologists, clinicians, we were able within 2 years to make a vaccine which produced immunity in animals and on challenge protected the animals. But sadly, Singapore at that time did not have the know-how technology and factories or pharmaceutical industries to scale up to make many millions of vaccines safely for the large number of people required not only in Singapore but for the whole world. Working with the International Agency for Research in Cancer, which had set up a cancer registry in Singapore, a national cancer registry in 1969, I and IRC and the World Health Organization in Geneva, with the aid of Dr. Perkins, who was then the Director of Biologics, decided to see how we could get vaccines and start a program in Singapore. WHO was very delighted that Singapore was proceeding to make vaccines and hoped that in one day, there will be vaccines for the whole of the region. I was given a $20,000 research grant from the World Health Organization of the International Agency for Research in Cancer to help with a project that would take me a lifetime to know the answer. The title of the project was, what was the outcome, or what is the outcome of liver cancer before and after hepatitis B vaccinations? This enabled us to get the best quality vaccine available and to move into the new technology of molecular techniques to make safe vaccines. Small-scale vaccinations were carried out using the plasma vaccine. In the early days, vaccine manufacture and the number of vaccines, but they were unsafe. We had the problem also at that time of the scare of the AIDS virus. At that time, no one knew what the infectious agent is, and we were making vaccine from infectious blood, where the AIDS agent may be present. So the toughest sterilizing agents were used to purify and clean the vaccine before it was ever used in human beings. We produced seal very slowly. The decision to use the vaccine in Singapore required the Singapore Cabinet approval because 100% of all our young people under the age of 20 would require to be vaccinated. And we could not afford to have a single disaster. It has been known in the past that in one accident involving vaccines in Canada using the Qatar laboratories, a vaccine, I think it was a BCG vaccine, was contaminated with sarcoma virus, and many thousands of children were infected with it. This book tells the story of the vaccination, the history of the hepatitis B virus, in the simplest layman's term, and written very well by my colleague Karen Kwek. It's a story of the search for a vaccine, how to make it, how stringent the safety methods are required, how scrupulously we monitored the vaccination in all our children and young people. At the time, in early 83s, we received only 10 doses of vaccine from a manufacturer. Not enough to vaccinate even the VIPs in the country. And the advice the advice of our Director of Medical Services, he suggested that our team used as handling highly infectious material, and for which many of our colleagues had died from being infected, should be the ones who should benefit from the vaccine. I, who was not immune, took the first shot, followed my colleague and my senior research technician, followed by the rest of our technicians. We prayed that we would not get into trouble from the vaccine. And God answered us, and we were safe, and many of us are well without any danger. Many years later, when HIV was diagnosed as the cause of AIDS, we took our sera and the blood of all those who were vaccinated with the plasma vaccine and tested it for HIV, and none of us were infected with that agent. We found some persons who were allergic to thimerosal and requested the manufacturers to remove it. Subsequently, all vaccines do not contain thimerosal, which is a preservative. The early plasma vaccines gave rise to a number of mutant hepatitis B virus, which were a serious threat to the vaccination. Fortunately, the yeast vaccine, which is made from Saccharomyces cerevisiae, yeast which is used in making beer, also was found to be useful for expressing the surface antigen from its gene, which was isolated. This could make copious amounts of vaccine safely. When tested subsequently, it was found to contain no mutants and no side effects in any of our vaccines. Since 1987, over 95% or close to 100% of our children have been vaccinated. In 2010, where this book tells a story, hepatitis carrier rate had dropped from 11% down to less than 1%. No child under the age of 17 is sick with acute hepatitis B, as contrast to the previous years in 1975 to 85, when there were so many sick children. And some of our medical students, or even some of our colleagues, had died from infectious jaundice when infected with this virus. Liver cancer rates have fallen from 28 per 100,000 in a population of 2.5 million in 1985 to 16 per 100,000 in 2016 in a population of 5.5 million. Less than 4% are infected with hepatitis B, and these are people who are migrants into the country. None of those who have been vaccinated required revaccination. The immunity is durable and lasting. Lastly, there is no liver cancer under the age of 40 also. The number of liver cancers in females has dropped from number 4 in the order of frequency to not within the first 10. And in males, it has dropped from number 1 position to number 4. And we do not see any familial liver cancers which occur in a young age group of under 30 years of age. This is all due to the benefit of the hepatitis B vaccination. Singapore was the spearhead of the vaccination program, monitored very closely by the World Health Organization and through our collaborating laboratories, in which Singapore was one of them. The vaccine in Singapore was also used in ASEAN countries, as well as in Myanmar. And many followed us, not only in vaccination, the adult population, but also learning from us that it was safe to vaccinate children from the time they were born, even combining it with other childhood vaccines like BCG or diphtheria, tetanus, polio vaccines, with no harm to any of the vaccines or any sequelae. In year 2000, at the WHO meeting on viral hepatitis and an review of the status of the vaccination program, nearly 15 years after vaccination has started, there were no disasters of hepatitis B vaccines or vaccination program, and it was going smoothly. That year in the Lancet, it was announced that the World Health Organization had requested or recommended that all endemic countries of the world should use the hepatitis B vaccine for their newborns, and those in non-endemic countries to use it in the early childhood or within the first year of the birth. In 2005, GlaxoSmithKline came to Singapore and started manufacturing vaccines, including the hepatitis vaccines, for all over the world. It was nearly 20 years after our first vision to set up a vaccine manufacturer in Singapore. This project not only provided the saving of millions of lives all over the world, but it was also the time of the transformation of industry. It was the Industrial Revolution into high technology, from a low-key technique to molecular and even involving other aspects of science and computers and scientific engineering. It's a transformation of the whole society, which also involve the schools, the colleges, and the universities. Many of the industries which you see today in Singapore is a result of that industrial transformation, which led to higher education for all the sciences, including medical sciences. This book is available, A Cancer Vaccine That Transformed Singapore and the World. It's published by the Straits Times Press Limited, and you may obtain a copy. The Straits Times Press book press is available on the internet. I wish you a happy reading. Hope you enjoy reading it. And read the story of the incredible life-saving work which was done by many thousands of scientists all over the world, collaborating together with one aim: to wipe out hepatitis B and liver cancer. Today, in year 2018, on the 22nd of June, 32 years after Singapore launched its vaccination program for all its population, we do not have liver cancers under the age of 40. Liver cancer has almost been wiped out. And now we have to deal with the treatment of hepatitis B. For we know that in our country, the major cause of liver cancer is hepatitis B. Although there may be other agents like hepatitis C or alcohol, but the predominant one is hepatitis B. I wish you a happy reading. May God bless you too.